Hello everyone, and welcome to Talking About Regeneration, the podcast that's about stem cells, gene therapy, and all things regenerative medicine, with perhaps some other fun stuff thrown in for good measure. I'm Kevin McCormack, the Director of Patient Advocacy at the California Institute for Regenerative Medicine, or CIRM. We're California's stem cell agency, and we have $5.5 billion to change the face of medicine as we know it. And this podcast will introduce us to some of the people who are helping us do just that. Enjoy the show. Our guest today is Dr. Dennis Clegg. Dr. Clegg is a professor of biological engineering and of molecular, cellular, and developmental biology at the University of California at Santa Barbara. And if you know Santa Barbara, you know it's a beautiful part of California, right on the beach, and it offers the perfect excuse to goof off. But no one could ever accuse Dennis of that. He's the recipient of the UCSB Distinguished Teaching Award, the Pacific Coast Business Times Champions in Healthcare Award, the National Eye Institute Audacious Goals Award. I love that word, audacious. He's been a Frontiers of Vision Research lecturer at the National Eye Institute, a keynote lecturer at the Stem Cells World Congress, and a TEDx speaker. There's a lot of other things on his resume, but this is not an hour-long podcast, so we don't have time to go into those. Dr. Dennis Clegg, welcome to the CIRM podcast, talking about regeneration.
Thank you. Happy to be here.
We're delighted to have you. So let's start with the work that you're doing. And obviously, some of this work is that CIRM is funding in the interest of full disclosure. Describe the work you're doing for macular degeneration and why it's important.
Yes, sure. Age-related macular degeneration is one of the leading causes of blindness in the world, and especially in the elderly. And there are two forms of the disease, the wet form and the dry form. And for the wet form, there are some treatments that work pretty well, but for the dry form, which is the most prevalent, there's really nothing you can do. And something like 3 out of 10 people over the age of 75 suffer from this disease. So we wanted to come up with a cell therapy that could treat the disease and prevent blindness in these patients. And it's a devastating disease where the very central part of your visual field just fails. And so imagine if you're looking out at a scene and somebody's holding a fist up in front of your face, and you just can't see anything in the middle of your visual field. So it's difficult to read. Of course, you can't drive. Difficult to watch TV, even difficult to recognize faces. Mm-hmm. So we, from previous studies by our groups and others, we knew that there's a cell type in the back of the eye called the RPE, or retinal pigmented epithelium. And that's the culprit here. Those cells, for unknown reasons, die. And they're support cells for the photoreceptors, for the rods and cones. And so once your RPE die, the rods and cones die, and you lose the ability to see in that central part of your visual field. So our strategy was to make the RPE from stem cells and implant them in the back of the eye via a surgical procedure to replace the dying cells with young, robust cells to keep the photoreceptors alive.
How's that going?
So with funding from CIRM, we were able to come up with a product, a cell on a scaffold that and figured out a way to implant it, working with the surgeons at USC and the engineers at Caltech. And then we went on to a phase 1 trial, and we implanted 15 patients, all of whom were legally blind. Mm-hmm. And the results, we followed them for a year, in some cases even 3 years, and the results are promising. 27% of the patients saw an improvement in vision of 5 letters on the eye chart. And to my knowledge, this is the only treatment that has shown such an effect for people that are legally blind. Now, 60% stayed the same or got better. And that's a good result because in the fellow eye, which serves as a sort of a control, everybody got worse and the vision slowly declined. So we're excited now to move on to a phase 2B trial, and we're starting to make cells for that trial under a GMP good manufacturing process so that we can move on. And we've been in contact with the FDA, and they're allowing us now to go into patients that are at an earlier stage in the disease. And we think there we'll even see better results because we can put the cells in, save the photoreceptors before they start dying.
All right. That's pretty exciting. I mean, to see not just kind of the disease not progressing, although obviously that's a big deal in and of itself, but to see it reversed, even if it's only just to say 5 letters, I mean, that's a remarkable thing to see.
Well, thank you. Yeah, I'm excited about it, and some of the patients are just over the moon after seeing their vision decline, and then all of a sudden it improves. And there's one particular patient who recorded a video online, and she talked about the fact that she didn't like to go to parties anymore. She couldn't recognize faces. She couldn't read. She couldn't watch TV. And all of a sudden, right after the surgery, she could see an improvement, and she actually met the team at an event at the International Society for Stem Cell Research, and she wanted to thank each investigator personally. And it
Wow.
was really heartwarming for all the folks in my lab that were able to meet her and attend that event.
That must be great because for so long, I think you work in a lab and you're working on cells, and it's all very important, but it's kind of academic, it's dry. And then to suddenly walk in and see this woman whose life you've changed, that must be so wonderful.
Right. One of my students said that patients and interacting with patients was like providing jet fuel for her to go back to the lab and work really hard. And yes, it's rewarding, and it makes us want to work harder and try to help people that have this terrible disease.
Yeah, we'll have to introduce patients to every researcher we fund so we can give jet fuel to all of them. So obviously you learn a lot from that first trial. So have you made kind of different changes, alterations, improvements in the delivery system, the use of the cells for this next phase?
Yes, it was a learning experience. To get to the first patient, we did studies in rats, we did studies in pig, but this surgical procedure had never been performed in a human before.
Hmm.
And the surgeons at USC, particularly Mark Humayun and Amir Kashani, were just amazing. They had to, feel their way through the surgical procedure in the first couple of patients, and they made improvements along the way. Some of the patients had some hemorrhaging in the first cohort, and they figured out how to prevent that in the second cohort. And so it was a learning experience all along the way. And we think we're ready now to go on to a bigger trial with more patients, where we hope we can see really good results.
That's exciting. And obviously we should give a shout out to the patients because they're pioneers in this. I mean, this is the first time it's been done in people. And so for them to be willing to kind of put their eye on the line, as it were, that's extraordinary.
Right. And these were patients in their 80s in most cases, and they were amazing. Yeah, they're like astronauts going up in the first spaceship, and their attitude was just so refreshing. They said, "It may not help me, but it'll help others. Let's go. Let's do it."
That's wonderful. That's so encouraging. So what are the biggest challenges that you and your team and other researchers like you face in doing this kind of research?
Well, it's a challenge because it's such a new technology, and not only is it stem cells and stem cell derived cells, but it's a novel surgical procedure.
Hmm.
It's not too different from what they do typically in treating diseases in the back of the eye, but there were a few wrinkles they had to figure out. So, for example, our scaffold that has the cells on it uh, rolls up into a little taco-like thing in a cannula that's then inserted into the eye,
Hmm.
and then it unfolds, and that allows the surgeons to keep the incision very small. And that took some engineering, some device manufacturing, and a lot of testing. The FDA wanted us to make sure that that folding didn't harm the cells. So we had
Right.
to take the device, put the scaffold in, extrude it, take it in, put it out, and then measure cellular functions. And we found that the cells were not stressed. So a lot of challenges with regard to the surgical procedure to make sure that the cells were going to survive the whole process and function.
Yeah, and also to keep the scaffold on the eye. I mean, you can't exactly just glue it or staple it on, can you?
Right. Well, there's pressure in the eye that pushes the retina against the back of the eye in a healthy eye, and that keeps the scaffold in place. And we didn't see it move at all. And one, really important question is, well, how are the cells doing after implant? Are they still alive?
Hmm.
And one of our patients passed away 2 years after the surgery due to other complications. And they had donated and consented to donate their eye to science. So we were able to look specifically at the cells. And after 2 years, the cells were still there. They were alive and functioning well. So, and this was a patient that had a huge area of geographic atrophy. They had hand motion vision only, so they couldn't see any letters on the chart. They could just see your hand
Hmm.
moving. And so we were encouraged by that. Now it's only one patient so far, but in terms of your shout out to the patients, most of them have consented to donate their eye after they pass, which allows for really definitive experiments to show that the cells are there and that they're still functioning.
Wow, that's such a generous thing to do. So on your bio page at UC Santa Barbara, it says it talks about the eye as complex, beautiful, and functional. So what is it about the eye that appeals to you so much?
Well, the eye is just amazing. I think Darwin called it the organ of perfection, and it's so important too. I mean,
Hmm.
it's part of the central nervous system that's right there and easily accessible, and and in a lot of ways, it's easier to study than some part of the brain that's deep inside the skull somewhere. And a lot of model systems have been used to study the development of the retina, and and it's so important to mankind. I mean, I've gotten involved with the Foundation Fighting Blindness and gone to events where there are a lot of blind people there, and just to to be able to try to help those patients is really rewarding. So not only is it fascinating science wise and biology wise, but it's also so important to try and treat these diseases.
Absolutely. So 10 years ago, and it's hard to imagine it's been that long, isn't it? You did a TED talk and you talked about learning how newts regrow limbs. And you said if we could understand how the newt does that, we could potentially figure out ways to do that in people. So how are we doing with the newts these days?
There's been progress made, and yeah, they're amazing. some newts, not all newts, but some newts can regenerate an entire leg if the leg is lost. And it turns out that that species of newt, the males fight each other a lot, and they bite off the other male's legs. And so they figured out a way to regrow the leg. Other species of newts can't do that, and it's fascinating. And imagine, I mean, that's a really complex tissue. It has nerves, it has bone, it has skin, it has muscle. And so they thought at first it was a pluripotent stem cell, like an embryonic stem cell or an iPS cell
Hmm.
that might be involved. But the work of especially Ellie Tanaka in Germany showed that it's actually multipotent stem cells for each tissue that are stimulated uh, in the case of an injury. And so there's a muscle progenitor that makes muscle, there's a nerve progenitor that makes nerve, and it all comes together. And again, fascinating science. We don't understand everything, but there's also been progress made in regeneration of the retina in the eye, which
Mm.
happens in zebrafish, for example, but doesn't happen in humans. And the work of Tom Ray and Dan Goldman has shown that there are certain transcription factors that are turned on in the glial cells, mular glial cells, that allow them to turn into photoreceptors and replace photoreceptors that are lost. And they've gone on now and tried using those factors in mouse and have made some progress in convincing the glial cells in mouse to turn into photoreceptors. So that's a promising way to, maybe via gene therapy,
Hmm.
to take existing cells that are still there and convince them to become photoreceptors to replace those cells that are lost in disease.
That'd be amazing, wouldn't it? To be able to kind of just kind of get your own body to kind of replace the bits that are dying off.
Yes, yeah. I mean, it's sort of an endogenous repair mechanism, and if we
Hmm.
can understand that, that might be a really great way to treat disease. Retinitis pigmentosa, for example, where you lose photoreceptors, but you still have the Mueller glia. So if you could convince them, go ahead, make a cone, make a rod, you'd be in good shape.
I like that. Convince them. Let's talk to them and let's make some sense. So, and one of the things I get when I go out and do public speaking is that people always ask, why does it take so long? I just watched a documentary about the search for a treatment for type 1 diabetes. And
Mm.
one of the woman who, the person who was making, excuse me, the documentary said the joke in the community is that the cure is 5 years away. It's always 5 years away.
Right. And I've given talks to patients and patients' families and for the foundation Fighting Blindness, and they've said the same thing. You guys, you always say 5 years, and now it's 5 years later, and you say 5 years. So, uh, and, and what I think is important to realize is that it's not easy to come up with these treatments.
Hmm.
If it was, we would have, figured it out already. Uh, the diseases are complex. In some cases, we don't know what exactly causes the cells to die, for example, and uh, and so it takes a while. I mean, even for development of a small molecule drug, they usually quote a figure that it takes 10 to 15 years, and it costs over $1 billion, and
Yeah.
90% of the molecules fail at some point in the development. So it's a difficult process, but again, we're driven to try to treat these diseases because there's a big need for it. It's unmet medical need. So we have to keep trying. And progress is gradual, but progress happens. And sometimes
Hmm.
there's a breakthrough where all of a sudden you've got a treatment. So we have to keep trying.
Yeah. How do you stay motivated then and hopeful? Because there are lots of failures along the way. I mean, every step you take, there's kind of a couple of things that don't necessarily work out.
Right. And when you're doing research, you're doing an experiment and you don't know how it's going to come out. And in a lot of cases, it doesn't come out the way you think. You're testing a hypothesis, and you're dealing in an experiment with, maybe 10 to 50% of the unknown. So that's what you're trying to figure out. And if it doesn't work the way you think, you go back to the drawing board and say, okay, that hypothesis was wrong. Why did that happen? Let's think of some new explanations. And you go back to the bench and do more experiments. And in clinical trials too, some fail and some succeed. And even if they fail, you learn something. You learn,
Right.
and it allows you to go forward with a new direction to try and figure out the best way to proceed.
Right. And so finally, what do you do for fun? What do you do to stay active and healthy? You're in Santa Barbara. You got so many options there.
Well, we're fortunate to live in a beautiful place, and we have a big blue ocean there, and I love to get out on the water and go fishing. I'm not a surfer. Everyone says, "Oh, do you surf?" I'm like, "No, I'm terrible at it." So, but love to go to the ocean and spend time there. And then we have beautiful hills here behind us, the Los Padres National Forest. And I'm a big dog aficionado. I've got 6 dogs in the house and love to take them out.
Oh,
And the
wow.
other thing I do that keeps me busy is I'm the president of our volunteer fire department. It is high fire danger here, and I've been active in uh, raising money for the group here, and also even learned how to roll up fire hose and put out fires. So that keeps me busy as well.
Yeah, sad to say it's become an increasingly common part of the landscape, isn't it? I mean, we're all kind of faced with that. Those of us who live in cities, less so perhaps, but certainly anyone in the countryside has to be a part of the solution.
Exactly. And we're trying to pioneer a sort of new approach where it's not just a group of 10 people that are the volunteer firemen, but it's the entire community that gets involved, that protects their homes. We have water sources around the neighborhood, pumps and hoses, and a number of folks that are trained and can put out fires if they come.
Great. Well, good luck with the firefighting and good luck with the research. Dennis, it's been great fun chatting to you.
Okay, thanks. Yeah, great to catch up, and best to you and all the folks at CERM.
Thank you. And I'd like to thank everyone who's been listening at home, in the car, or in the shower, or wherever you take your podcasts. And we'll see you next time on Talking About Regeneration.